wonderful introduction. I hope today, well, first off, I apologize if I trail off in the middle of a thought because I'm quite heavily jet lagged and my body currently thinks it's like 1 in the morning. So I may have some slips. The other thing I wanted to say is that as I launch into this, what I hope to offer today is rather than kind of an overarching story that I would normally do, I'm hoping to offer sort of a smorgasbord, or in my part of the world we call it a buffet, which I think is a French word. So half the words in English we stole from French. So an offering of some of the stuff that we've been working on in our group, and that hopefully can offer some opportunities for collaboration as I'm here for at some point over the next year, 2 years, maybe permanently. So I'm probably going to be speaking about the role of the immune microenvironment and how we can use this in our routine care of patients. As Vera said, so I worked with Pierre Messiaen. So this picture of Pierre was made by a radiologist at Vanderbilt who went to PubMed and scraped every publication that Pierre was on, and then captured all the names of the co-authors, and then created this portrait of him from the names of the co-authors. So I think several of you are co-authors with him, and if so, you can probably find your name in here as well. My name is here. Oh, she said, don't touch that. It might just. So Pierre Messiaen was a visionary and inspiring figure who he always made sure that everything that we did was inspired by patients. And in fact, what he would start all of his meetings with is that as a group, we want to be inspired by patients and driven by science. And so inspired by patients means that everything that we do, he made sure had some connection to how this was actually going to improve patient outcomes. And so everything that we did, when we brought an idea to him or a new analysis of data, he would say, okay, this is cool. Whoop, this is cool, but if it doesn't actually have a direct translational impact to something that we can do to actually help patients in the future, then it's not worth our time. This ranges from things like in our lab, we do a lot of clinical implementation science. We're looking at ways that we can use biomarkers to help inform patient care. But then also to the basic biology side. I mean, Pierre was a great basic biologist. We did a lot of genomic work. We're doing immune microenvironment work. So it doesn't have to be something that's definitely clinically translational just yet. But as long as there's a link to how, thinking forward, how can we use this to guide patient care in the future? So really quickly, I don't need to spend much time on the rationale for why lung cancer. At least in the US and worldwide, I don't know exactly in France specifically, but lung cancer is the deadliest in both men and women. And it's the deadliest cancer, and it's deadlier than cancers 2, 3, 4, and 5 combined. And then a lot of people think that cancer is a disease of smoking, and smoking does greatly increase your risk of cancer. But at least in the US, we're starting an initiative to look specifically among lung cancer among non-smokers. And I think that this is going to have a huge impact on how we treat both cancer in non-smokers and in smokers, because a lot of the risk factors, if we can find them in non-smokers, that will better guide or inform how we manage people who have smoking aggravated lung cancer. So among non-smokers, lung cancer would still be the 5th deadliest cancer if it were its own classification. So the burden on healthcare is huge. The 5-year survival of lung cancer is less than 20%. Actually, this was just updated last year according to the US SEER database. This has increased to about 22%, which is incredible. We're actually making progress over the past decade. We've increased survival from like 15 to 22%. So I mean, that's like a 30% relative improvement. That's huge. One of the biggest problems with it is we don't normally detect it until late stage, because if you become symptomatic, it's usually much too late. So we can find it through screening or incidentally detecting nodules, which then we have to decide how we manage them. So we know that early detection saves lives, and screening programs in the US and in many other countries are growing. I saw that there's not a unified screening program here. So maybe that's something we can work towards. That's okay. I mean, we have lung cancer screening in the US. Anyone who's over the age of 55 who has smoked more than 15 years in the US is recommended for lung cancer screening. And our Medicare, which is our semi-public healthcare coverage, it's complicated, does reimburse screening for that. However, of those eligible for screening, only 5% actually get screened. So while we do have lung cancer screening, it's basically non-existent. So nodules, which could be lung cancer or maybe not, are commonly detected on CT. And then we have to decide what to do with these patients. So the focus of our lab over the past decade or so has been to help manage patients with a nodule. And so a lot of people think a nodule is sort of this nebulous thing. What is it? It's a spot in the lungs that when you look on an X-ray or a CT scan, looks like it could be, something that's not normal lung tissue. And a great analogy for this is think of moles or freckles. I do have some Italian heritage, and that almost entirely comes out in having lots of moles all over my skin. But it's like a mole in that, most moles are benign. There's nothing wrong with them. They're not going to do anything to you. And occasionally one might actually be skin cancer. And so that might be worth excising or just keeping an eye on. The problem is the lungs are a bit harder to reach than the skin. The skin's on the surface. If there's anything that looks slightly suspicious, you can just cut it off, no problem. You wear a Band-Aid for a couple of days. That process is a bit harder within the lungs. So when we find something in the lungs that's suspicious, we hope to be able to develop a way to non-invasively determine if that is worth taking a closer look at or not. So right now, when a patient comes in with a lung nodule, and in the US, this rate is about 1.6 million lung nodules are detected in the US every year, not through screening. So these are people who come into the emergency room with a broken rib, and they get an X-ray or a CT scan, and then by the way, you've got a lung nodule present. Or patients that had some sort of, they had some other treatment they were undergoing, and so as part of their preoperative evaluation, they got a CT scan, and now you have a lung nodule. So let's look at it more closely. So among screening detected patients, this number is skyrocketing. So we have millions and millions of people every year that have these nodules, and we have to decide what to do with them. Are they cancerous? If they are cancerous, are they going to be dangerous? Do we need to take them out? Do we need to take the patient's chemo? And so the first step here is we apply a clinical prediction model where we look at factors such as the patient's age, how much they've smoked, how big the nodule is, some other features from the CT scan, and we can say, okay, some of these patients are low probability of cancer. And these patients will send them to CT follow-up. Come back in 3 months, we'll look to see if the nodule has grown, and if it has, then we'll do something else. If not, then we can safely say that this is a benign nodule. The high probability nodules will send straight to surgical resection, especially even if it's not cancer, if it's a very large nodule, if it looks like it's growing, if it's some sort of inflammatory process, sometimes it's better just to take it out anyway. So we'll send them to surgical resection. But this intermediate probability group are the patients that we're not quite sure what to do with. And a lot of these end up being cancer. But the really interesting thing is that a lot of them might be cancer that won't actually harm the patient. There are some forms of adenocarcinoma, which I'll get into in a second, which we call them indolent. It means they're not going to grow. They're not going to actually harm the patient. Or if they do, it's going to be years down the line. And so if you have an 80-year-old patient with an adenocarcinoma that might not grow and harm them for another 10 years, it's probably not worth taking them to surgery and cutting out an entire lobe of their lung. So this intermediate risk group are the patients that we aren't quite sure what to do with. And so we hope that by adding a biomarker, we can better rule out some of the low probability patients, better rule in some of these high probability patients. And then among this group, we can decrease the number of unnecessary procedures, and while also reducing the time to diagnosis for patients that actually have cancer. We know that after about 90 days of waiting, there's about a 1% chance that cancer is going to shift positively in stage, meaning it goes from stage 2 to stage 3. And that can have a huge impact on the survival of the patient. So this used to be an hour-long presentation that I'm going to condense into this one slide. We spent the past 10 years or so developing biomarkers to look at, okay, we have a patient with a nodule. Can we decide if it's likely cancer or not? What do we need to do about it? And so we developed what we call a combined biomarker model, where we developed a blood test. We developed a radiomic signature where we looked at quantitative features of the nodule in 3 dimensions on the CT scan. And then we also looked at expanded clinical history, information about the patient's behavior, their health history, stuff like that. And we added all these things together, and we showed that, hey, we're more accurate than standard clinical prediction model. And we didn't have to do anything invasive. We don't have to take a biopsy. We don't have to put the patient under anesthesia. Actually, in the US, we like to take people to bronchoscopy. Bronchoscopy is you would have to go under general anesthesia. But I actually just learned that in the UK, they do all this under like laughing gas. Here, do you send people to bronchoscopy in general anesthesia, or is it? So it is kind of a laughing gas and going into the, okay. See, it's really interesting because then that kind of negates a lot of the challenges that we face. And when I tell people in the US that, well, I don't think other countries do bronchoscopy the way that we do. Oh, that's terrible. I wouldn't want to be awake, for someone sticking a tube down into my lungs. And then when I talk to colleagues from the UK, and it's like we put people under anesthesia, they're like, why? Just tell the patient to sit still for 10 minutes, stop complaining. Love it. I love the small cultural differences between the US and Europe. Like in the US, we allow people to buy assault rifles. That's a different story. We already talked about it. So we developed this non-invasive method, which I guess for the people who don't undergo full anesthesia, this would save them from having a tube stuck down their throat while they're awake. So probably would be helpful here as well. But so the question is, just changing the patient's probability of cancer isn't necessarily a patient-centered outcome. And again, in our lab, we're always focused on making sure that what we do is inspired by patients. So, okay, we can more accurately predict whether it's cancer or not, but is this actually going to have an impact on what we do? So in the US, guidance for patients with nodules is based upon these risk groups. So we have a no test or a rule out threshold, which is lower than 10% probability of cancer, and a rule in threshold, which is 70% probability of cancer. Rule out, if you're below that, then you just go to CT surveillance. Rule in, if you're above that, you go straight to surgery or possibly the oncologist. In the intermediate risk group, we're not quite sure what to do. So each dot here is a patient. The x-axis is their risk based upon the clinical model, and their y-axis is the risk based on the combined biomarker method that we've developed. And so for patients that ultimately want to be benign, any dot here that's in gold is a patient that we successfully ruled out. So these are patients that we could have taken from the intermediate risk group, moved to the lower risk group, and then could avoid an unnecessary invasive diagnostic procedure. Same thing for cancer. These are patients that we moved up appropriately. The interesting thing here is we then went back and looked at, okay, what are these patients that we got wrong for benign that we would have sent straight to surgery? And what are these cancers that we got wrong that we would have delayed their care? We went back and dove into these patients' medical history, and we actually found out that all of these were benign, indolent cancers. So even though our biomarker incorrectly reclassified them, these are patients that we wouldn't have actually harmed by delaying their care. In fact, this one down here in particular, that was an indolent adenocarcinoma that did not grow on imaging for about 6 years. And then after those 6 years, they ended up going in for a different procedure. While they were in, they took it out just because they were there. And that was when it was actually diagnosed by pathology as an adenocarcinoma. So this wouldn't have actually harmed the patient or probably impacted their quality of life. So anyway, so we've been developing these biomarkers to decide what to do with patients once they have a nodule. Yes. But then that gets me to the idea of what can we do with something that's likely to be a cancer and decide whether we need to actually treat it. And so I am a huge Star Wars nerd, so I wanted some way to kind of take my project and put a Star Wars reference to it. So if you've watched The Mandalorian, the armor that the Mandalorian wears is made out of a special type of metal called Beskar. It's like the strongest metal in the galaxy. It can stop lightsabers. It can stop lasers. And so I'm calling this biomarkers for early stratification of lung adenocarcinoma recurrence, because a lot of these ideas of whether a cancer is going to be aggressive whether the cancer is going to invade the surrounding tissue, if it's going to metastasize, if it's going to grow fast enough to kill the patient. What that really translates into in terms of a patient-centered outcome is the cancer going to recur after we treat them? And so using recurrence as the surrogate endpoint for aggressiveness, or the other way around, aggressiveness is a surrogate endpoint for the patient-centered outcome of recurrence. So this was a really important landmark study that came out about a year ago that showed that actually doing neoadjuvant immunotherapy and chemotherapy provided better survival to patients with early stage adenocarcinoma than chemotherapy alone. Now, this is another great thing about working in lung cancer, is the fact that like neoadjuvant therapy has been pretty standard in most other cancers for like years and years. And we're so far behind that like all we have to do is replicate what other cancers have already done. And for us, it's a huge win. So it makes it a little bit easier. We can just copy paste, and then we get to feel good about improving patient survival. But so the idea is that giving patients neoadjuvant therapy improves their survival. And now the question is, who are the patients that we need to give the neoadjuvant therapy to? Who responded and who didn't? Who are likely to have a recurrence or failure to go into remission, and who's not? And can we better guide who should get these therapies based upon that? So this is where we get into the clinical problem. At the current time, there's no biomarker determining which patients are likely to undergo recurrence. Right now, we look at it based upon whether the patient's stage. We look at a number of genetic mutations present in the tumor or not to decide which treatments to put them on. But right now, there's not a unified way that's very accurate to say this is an aggressive cancer and we need to treat it aggressively versus this is an indolent cancer that we can probably leave alone. So the idea as part of this project that we're developing is that can we identify biomarkers, again, non-invasively, to better inform how patients need to be treated. And so this is an example of the timeline of patient care and showing how currently the way that we manage patients, we're really putting the cart before the horse. So we detect a nodule on a CT scan, then we do a diagnostic bronchoscopy. I guess this actually would be the European style because this guy's sitting up. If it was in the US, I guess he actually would be lying down. But so then after the diagnostic bronchoscopy, we would start neoadjuvant treatments. Then we take a resection following a month or 6 weeks of neoadjuvant treatment. And then from that resected tissue, we get the pathology to actually fully grade the tumor and inform care. And this is going to decide whether we think how aggressive it is or not. And then we can include adjuvant treatment, follow up, how many times are we going to evaluate this patient by CT imaging or repeat biopsy, whatever we need to do. The problem here is this is the information that tells us how aggressive the cancer is going to be. This is when we need it. And so hopefully using imaging data, using tissue from the bronchoscopy, and using blood biomarkers, we could better predict the aggressiveness, but we would have this information here so that we could better guide the patient care. So that's kind of the overarching idea. So right now there are a lot of biomarkers looking at aggressiveness, survival, recurrence, but none of them have actually been put into clinical use. And so one of the issues is lack of availability in the clinic due to the cost or the complications. So one of these is multiplex immunohistochemistry. We actually just had a study I'll show in a second using multiplex immunohistochemistry, which very accurately predicts recurrence-free survival. But the problem is multiplex immunohistochemistry is it's a laboratory technique. It's expensive, it's complicated, it fails a lot. And so having that as part of routine patient care is not really something that's going to be helpful. Whole exome sequencing is another one. We've got some great data looking at sequencing the entire exome of the tumor, finding all these mutations, doing correlation analysis. This is a little bit too expensive to really do in a clinical space. Now in the US, we'll probably find a way to pay for it because that's what we like doing. If we can throw more dollars at the situation, well, I know in Europe that's, there's actually other considerations that go into it. And then the other thing is there's a lack of actionable information because we focus on aggressiveness, But aggressiveness, again, is not a patient outcome, and aggressiveness is not a direct one-to-one indicator of what we can do to better inform that patient care. And then lastly, as I showed you before, this information that we have is available after it's needed. So, yes, again, with the Star Wars references, the great thing about saying that hindsight is 20/20 is that Yoda would be able to say it the same way, you know, forward or backwards. So always 20/20 hindsight is. And so the question is, why are we well equipped to do this, and why should we embark on this now? So at Vanderbilt, we've been developing, as you've seen from our earlier slides, radiomics analysis. We've been developing tissue-based analysis. We've been developing blood biomarkers, and we've been developing ways to integrate them all together. And we have the patient information, and we have collected specimens. Now, what I think is going to make a perfect match between what we've been doing at Vanderbilt and what's going on here in Toulouse, especially with Vera's group, is really refining this integration step. Because when I say we have expertise in integration at Vanderbilt, we're doing, simple machine learning, we're doing logistic regression, we're doing lasso, we're doing support vector machines. But our student who came here, Mafe, well, she's now Dr. Senosian. She really dove deep into the best ways to really integrate all these data types to see sort of the larger picture. And so that's what we really need at this point. It's because we have all these pieces of information. They each kind of show us some of the picture, but we need a way to really integrate it all together. So I think that's where, I think this collaboration that we hope to foster can really excel. So real quickly, talking about blood biomarkers. We've been looking at radiomics, genomics, blood, and the immune profile. And so I'm going to go into each of these in a little bit of detail. So we're looking at focusing on the pragmatic question, because again, patient-centered outcome. Can we move this into the clinic? What are the actionable decisions that we make? And so we hypothesize that a combination of radiomic tissue and blood biomarkers can improve the prediction of recurrence after surgical resection of early stage lung adenocarcinoma, which can then be used to inform aggressiveness of neoadjuvant treatment or resection. So here's some of the data that we have. So first, I'm going to talk about this radiomic tool that we developed called Canary. So my mentor at Vanderbilt, Dr. Maldonado, who is himself a Frenchman, grew up on the southern coast. It's a small town east of Montpellier. I don't remember what it's called, but whenever I told him I was interested in moving to France, he said, "Oh, but the French, they're also pessimistic. They just sit around and complain about things all day." And I was like, "Fabian, have you had a conversation with me? That's what I do. These sound like my people." But so anyway, the Canary approach, what they did was actually many radiomic approaches look at finding features and then correlating those features with an outcome. Here they went the other direction. They did an unsupervised clustering using affinity propagation and found that that adenocarcinomas on CT image cluster really nicely into 3 different groups. And then when you look at the survival of these 3 different groups, they split beautifully into least aggressive, indolent, medium, and then very aggressive cancer. And so the patients with a high Canary score had very poor survival. So this is one we're in a pretty good spot. So from imaging information alone, we can predict pretty well how aggressive the adenocarcinoma is going to be. Now at the Mayo Clinic, they're actually using this within clinical practice. This is not actually it's not fully FDA approved. It's not recommended by guidelines, but it has found some clinical use and being helpful in guiding patient care. Let me see. Yes. Okay, so that was one approach to radiomics, unsupervised clustering, finding these different groups and then showing that they actually correlate with outcomes. The other way that we've been focusing on is by taking and extracting radiomic features. Is there an imaging group here that does CT imaging analysis sort of stuff? Okay. I'm going to take your silence as the fact that yes, you need to hire me to come. You do? Okay, great. So the idea behind radiomic analysis is that we can actually extract quantitative values based on the three-dimensional distribution of the tissue as detected on CT scan. So in the past, the way that this has always been done is a radiologist who's been trained to read CT scans looks at the scan and then grades whether they think it's likely to be cancer or not. And also, it's wonderful. If you ever get the chance to read a radiologist report, it's like reading a different language. It's like looking at it's almost like the figures that they're using are actually from Arabesh, which is the Star Wars alphabet. It doesn't make any sense. They're barely actually using real letters, and it's completely indecipherable to anyone who's not a radiologist. But for them, it's a very efficient way of saying that, okay, this is very suspicious, this is not suspicious, this one should be followed up, this one's fine. But that doesn't give us something very quantitative that we can then use to build into predictive models. So what we do is we look on the CT scan and we find, so for example, this is what the nodule would look like on the CT scan. This would be healthy lung. Well, I say healthy, there's a bit of emphysema here. Oh my goodness, there we go. Don't touch it. Here we've got some airway and some vasculature, but that's not normal. That's a nodule, probably cancer. We then take this nodule and then we can segment out the region that is nodule in 3 dimensions. There are various tools to do this. There's some automated ones, there's some manual ones. Not quite so important. Those are all, smaller sub-projects. But so we segment out the nodule in 3-dimensional space, and then we calculate all sorts of features that describe that mathematically. So things ranging from its size and shape. So what's the diameter? What's the volume? How is the shape distributed? Is it round? Is it flat? Is it rectangular? We look at the texture, which is how is the density within that region distributed? Is it homogeneous? Is it heterogeneous? Is the heterogeneity homogeneous or heterogeneous? All of these things can be captured based on texture. And then we also do higher order transformations of the image, and we can look at things such as wavelet transformation. So to me, this was a nebulous idea at first, but then I realized that it's very similar to taking Fourier transform of an audio signal. So if you have a stereo and it has the little bars that bounce in time with the beat, and the ones on the left bounce in time with the bass, and the ones on the right bounce in time with the vocals or the guitars or whatever. That is taking an audio signal and converting it into a spectrum. We do the same thing with images in 3 dimensions. The math is almost the exact same. I guess here stereos have the bass on the left and the treble on the right. Is that? You look at it, I don't know. Who knows? Again, cultural differences. So when we look at these radiomic features, we can also split them out. We can also predict better survival versus worse survival. Now, interestingly enough, this particular model is not trained specifically to look at aggressiveness. This was a model trained to predict cancer versus no cancer. But we find even in cancer versus no cancer, that score can predict survival within cancer. So it can predict indolent versus aggressive. So, a very low-hanging fruit for this project is to then take the same data that we have generated on adenocarcinomas and then look at that in terms of predicting aggressiveness. The other thing is you're going to say that, okay, well, you've already predicted aggressiveness. Why do you need to do it again? And so these radiomic features predict survival. And then we looked at, well, what are our radiomic features telling us versus what is canary, which is the unsupervised clustering analysis of it. And we find that there are some features that correlate well with the canary score. There are some features that correlate well within a certain range, but then do not correlate in a different range when compared with canary. And then there are some that correlate hardly at all. And so the idea here is that the radiomic features and the unsupervised clustering are not telling us the same thing. So we can integrate this data, hopefully to improve the prediction performance. All right, so key takeaway here, radiomics, we can predict some good stuff. We've got a great starting point. One of the huge benefits to a CT scan, using that to predict performance and guide patient care, is you don't have to do any other procedure to the patient. The patient has already had the CT scan because that's where we found the nodule, and we said this is likely to be cancer. So all we have to do is then apply an algorithm. Algorithms are really cheap compared to any sort of chemical or molecular analysis. So then let's talk about genomics, because radiomics is going to get us partially there, but there's a lot about the biology of the tumor that can inform the care that we might not have. And so this was actually some of Pierre Massion's work. Dr. Senosian also contributed to this, looking at how the genomics of the tumor are different in patients who had an indolent versus an aggressive tumor. And so they did it, and they found that when looking at adenocarcinoma in situ, which is an adenocarcinoma that looks like an adenocarcinoma, smells like an adenocarcinoma, but doesn't actually invade the surrounding tissue like a full-blown aggressive adenocarcinoma, had a different genomic signature than minimally invasive adenocarcinoma, which then also had a different signature than full adenocarcinoma. These are representing these different gene sets. So there is some information there in the genomic data that we can use, and that makes a lot of sense. I mean, come on, the biology of the tumor is hugely impacted by the genomics of the tumor, and so if we're getting this tissue from a biopsy, we should be able to find a good way to incorporate some of that genomic data into the prediction of how aggressive the cancer is likely to be. So the takeaway here is that genomic analysis of the tumor also predicts survival. We also, so the previous data was targeted sequencing. We also find similar things when we look at whole exome sequencing. Now the signals here are different. Whole exome sequencing, we're looking at the entire exome, which means that our reads are not quite as deep. Targeted sequencing, we're looking at specific genes and sequencing them very deeply. And so whole exome sequencing allows us to take a very wide view, while targeted sequencing allows us to really focus in on specific genes. The signatures are different because what is significant in targeted sequencing, those genetic mutations might not be prevalent enough to be picked up when we don't sequence quite as deeply as is possible in whole exome sequencing. But on the other hand, whole exome sequencing, we can sequence many more genes, and so we can build larger panels to be able to understand. So there may be some complementarity to using these two approaches. So let's do deep sequencing, targeted sequencing on 1 or 2, maybe 10 genes that we know are highly predictive, and then we can do whole exome sequencing on everything else to incorporate in some of that sort of data. So again, key takeaway here is that there are specific gene mutations that predict survival, and then also signatures based upon whole exome sequencing. I know I'm blowing through a lot of the specifics here. I haven't mentioned any of the gene names. We can discuss those at length if anyone is really interested. That's again, not the purpose here. I'm trying to offer a buffet. I'm not offering a well, in the US we have Michelin stars. Do you have Michelin stars here? Michelin star restaurant giving you the best, most exquisite type of food from a single author. So then the other thing is that there is information in the blood that's correlated with the tumor and whether that's highly predictive of survival. So interestingly enough, there's been a ton of blood analysis looking at blood tests that have looked at the genomics in the blood. They have been looking at circulating tumor DNA, cell-free DNA, fragmented DNA, and then DNA methylation that's in circulation. And those have all proven to be pretty good and pretty accurate at looking at the differences between outcomes. The problem with all these genomic analyses, in my point of view, is that they're just much more expensive than a standard protein assay. A blood assay using a standard ELISA for something that's currently being run in the hospital clinical lab. Something I think Wander just dripped on it. Okay, that's fine. I just want to make sure it's hallucinating. Doing the juggling. So the this is the equivalent of it costs about $15. So that's what, €13. Is the cost of one of these blood tests in the clinical lab. So if we can find 1 or 2 proteins that are also informative of survival, that are complementary to the CT scan that we already have, that could lead to really quick implementation of sort of a low cost added value sort of test. And so this is a panel that we worked with with Abbott to develop for lung cancer prediction in nodules. But we found that there are actually some of these blood proteins that are very highly predictive of survival among adenocarcinoma, particularly SCC, ferritin, and CRP. The great thing about CRP is the CRP assay already exists in like every single hospital and clinic, even like small rural clinics run CRP because it's, it's a great inflammatory marker. It has huge implications for, heart attack, so if we could find something, a signal that was present somewhere here, we could move it into the clinic really easily, really quickly, and for very low cost. And so, I think that the genomic approaches to blood tests have a lot of merit, and there's going to be a huge future for them. But in terms of, again, what are the pragmatic steps that we can take to take what we've learned about the biology of the tumor and translate it into a clinically actionable sort of approach? I think this is, kind of the lowest bar that we can jump over. Another great thing, as I've said before, in lung cancer, we're so far behind all the other cancers. A lot of these markers are currently in use for other cancers. CEA125 and AG4 are currently in use for ovarian cancer diagnosis in the US. CIFRA is currently available in China and maybe here. It's not in the US, but I think it's in Europe for lung cancer. CEA is used in several solid tumors. So, these are things that are already currently. So again, key takeaway here, circulating blood biomarkers predict survival. I think we're seeing a theme here. The other thing is that there are companies that are developing specific tests to look at risk of recurrence, but then also the immune system's response. And so conflict of interest disclosure, I do consulting for Biodesics, this company, but I'm presenting published research. So, hopefully you will not perceive any sort of conflict in me presenting this. They developed this test called Veristrat. And what they did here was they took, they did mass spec on whole blood. And they found all these signatures. And then they again use unsupervised clustering to find signatures within the mass spec of the protein within the blood. And then they correlated this with outcomes. And what they found is that there's a signature, which they're now calling Veristrat, which is indicative of the body's, essentially the status of the body's immune system. They actually, up until recently, never even looked to see what those proteins were, but they found very high correlation between the outcomes, such as recurrence and progression, and the values of the test, where the test did not specifically correlate with any sort of biology that they could detect within the tumor. So again, Veristrat's not showing us what the tumor is doing; it's more showing us what the body's immune system is doing. And so I think that's a really interesting approach because, again, a huge part of what we understand of the biology and what a lot of the research here is focusing on is that, there's so much more to our ability to control cancer than just the tumor itself. It's the immune microenvironment that has a huge role in it. We've also been developing a high sensitivity assay for CIFRA-21-1. This was what my PhD was developing this technology, and this allows us to look at some of these same protein biomarkers at very low concentrations. And when we do that, we can be more accurate when diagnosing cancer or not. So this allows us, this is the ability to develop assays using a new technology that's cool. Everyone loves new technologies. They're fun. They're also a huge headache because then when they break, you can't just call the company and say, hey, come fix it. You gotta fix it yourself. But this is an opportunity to develop new assays, look at new protein markers sort of stuff. So key takeaway here, circulating blood biomarkers predict survival. So then we can talk about adding all these together to look at indolent versus aggressive adenocarcinoma. So we know that early stage lung adenocarcinoma can present with indolent and aggressive. We know that immune cell interactions play an important role. And so this leads us to our research question: how does the immune microenvironments influence recurrence? So I want to talk here specifically about some of the data that we've looked at when using multiplex immunohistochemistry staining. And I'll be honest, the whole reason I got into this project initially was before I got into lung cancer, before I even went into biomedical engineering, I wanted to be an astrophysicist. In fact, my undergrad was in physics. I've always loved astrophysics. Maybe that's some of the reason I love Star Wars so much. And so when looking at like images of exoplanets, I was like, these are the most gorgeous images possible. You know, artist representations, what exoplanets might look like, alien life and all that kind of stuff. And I was so jealous that these other fields got all these cool images. And like in the biomarker space, the coolest images that we get are Kaplan-Meier curves. So when I saw this data, I was like, Pierre, please, can I work with this data set? This is gorgeous. It looks like we've got a bunch of different planets, you know, something like that. And he was like, yeah, sure. We have all this data and we need to do something interesting with it. So what this is a tissue microarray, which we make by taking resected tumor specimens from a bunch of different patients. And we cut out a little punch from each patient, and we can line them all up on an array so that we can stain these with a bunch of different protein markers and look at them side by side. This allows us to completely eliminate any sort of batch effects that come from the staining process. If any of y'all have done something with immunohistochemistry staining, getting consistent stains from slide to slide is its own challenge. And if all you're looking at is the presence of one or two markers, that's usually fine. But if you're looking at a panel of markers, this can lead to huge problems and variations of batch effects, especially when you're trying to get really quantitative. So we do a tissue microarray. We can stain it for a bunch of different markers. Here we stained it for B cells, T cells, T regulator cells, hand-sided keratin, which detects cancer cells, mast cells. We're looking at DAPI so that we can identify nuclei. And then we've got a staining for proliferation status, which is Ki67. So based on all of these, we can look at what are the proliferating cancer cells, what are the proliferating immune cells. And when we look into these data, we can then, from this image, look at which markers are present and use that to identify and spatially resolve each cell. So each dot on this plot is a cell that's been identified from the staining patterns from the slide. And then based on the distribution of the individual cells, we can decide what is cancer, what is stroma. So what's tumor and what's stroma. And so what we found here is that it's actually not quite as important how many immune cells are present. That is important, but it's how many immune cells are present and within the tumor tissue. And so here are two slides. This is the most egregious example I could find because you know, when you're not able to look at the slide closely, maybe all you see is some blurs of color. But so we have so one of these is a patient who went on to have a long survival with no additional therapy besides their initial resection. And one of these is a patient that went on to have a recurrence even after surgical resection and adjuvant chemotherapy. So quiz for the audience, which one is which? This one is the one that had the recurrence. And you can see that the pink is one of our immune markers. And you can see that there's, you know, this is an immune desert. This is an immune hotspot. And so we found that there was this very strong correlation between the density of different immune cells in patients that had a recurrence versus patients who had no recurrence. So that's cool and all, but that itself is not necessarily clinically useful. Can we predict their actual survival from this? So we did find that there's a survival difference in early stage adenocarcinomas when we split groups based upon the density of mast cells. And mast cells are really interesting because if you look in the literature, if you read 3 papers on mast cells, you're going to find 5 different opinions on their role in in cancer development. And so if you, one of the other great things about mast cells is if you want to find that mast cells increase cancer proliferation, there's a paper for it. Decrease cancer proliferation, there's a paper for it. Modulate the immune pathways, don't have any interaction with the immune pathways. You know, improve survival, confer worse survival, whatever it is, mast cells are involved. So we wanted to look at the mast cells a little bit more deeply. And what we found was that mast cells specifically within the tumor tissue, not just present in the punch and the resected tissue, but within the tumor tissue itself, was very highly correlated. So the patients with the patients in the highest 25%, 25th percentile, sorry, the jet lag's here, had very good recurrence-free survival, whereas patients within the lowest 25% had very poor recurrence-free survival. So the density of these mast cells, not the characteristics of them, not the density of mast cells within the total tissue, but density of mast cells, specifically within the tumor tissue, was very highly predictive of recurrence-free survival. We found similar things for T cell density. Again, T cell density within the tumor. Now, T cells aren't quite as controversial as mast cells. So I don't think this was quite as surprising, but T cell density within the tumor, high density conferred very good recurrence-free survival. And then once you were out of that highest probability or highest density group, survival dropped off. And so again, the idea is, can we translate this to clinical practice? Because multiplex immunohistochemistry, as I mentioned before, is an expensive process. It's tedious. It fails a lot. There are a lot of times when you go to do your experiment and it just doesn't work. Things don't stain. If anyone's actually had to do multiplex immunohistochemistry, I'll pray for you. We outsource it all to collaborators because then it can be their headache. But so the thing is that CD3 is already a commonly used stain within our clinical pathology labs. I don't know if it is here, but in the US. They already use it, and they use it for several other cancer types. So we can already look at T cells. And so we went ahead and had the same tissue microarray. We just sent it down to our pathology department, had it stained with their clinical assay. And we're currently working on seeing if we can extract the same sort of information on T cell density within the cancer cells. Because again, if that's the case, these punches are 1 millimeter in diameter. The needle that the bronchoscopist uses when going in for a tissue biopsy is between 1 and 2 millimeters in diameter, depending on which needle. And so that means that from the same punch size, we can get the information that on the status of the immune system within the tumor itself. And then we can stain it with a currently clinically used assay. Now, this image costs like $40,000, so 35,000 euros, something like that. This one costs 100. So in terms of pragmatically being able to implement something into clinical practice quickly and effectively at low cost, I think we have a real opportunity. I also want to show, again, the question of mast cells. I wanted to show, this is actually some of the data that Mafe produced while she was here, where you probably are quite familiar with this image. So she did single cell RNA sequencing of 15 resected lung adenocarcinomas. There ended up being 6 indolent, 7 aggressive within this group. And so when doing a UMAP distribution of all of these cells, we find that this is the mast cell island. And then when we look at patients who are intermediate, indeterminate, and aggressive, almost all of the mast cells were present within this indolent. There's some mast cells in the intermediate and some in the aggressive, but almost all the mast cells are here within this indolent group. So there's something going on biologically here. And I think that there's a real opportunity to untangle what that is. I don't know how to do all this, so I'm hoping one of y'all is interested and wants to help and collaborate. I just think these make pretty pictures. That slide was out of order. No, it wasn't. Sorry, I'm going to blame jet lag again. At some point, this is incompetence, but I'm going to keep blaming jet lag. So then we wanted to look at, okay, so what's different about these mast cells? And is there a way that we can correlate this with some of the other biology of the tumor? So we actually found that we had a data set where we had 73 resected adenocarcinoma tissues, where we had this tissue microarray analysis to look at the density within the tumor tissue. And then we also had whole exome sequencing analysis, so we can look at specific mutations that were present. And the first thing that we found was that, yes, there were some mutations specifically that had a difference in mast cell density within the tumor. Here's a list of them. At the end of the day, I'm not a basic biologist, so this all kind of looks like alphabet soup to me. And again, maybe someone here actually recognizes something interesting here. The numbers here are how many patients were had that specific mutation. Now, specifically looking at a couple of these, MUC3A and MUC5AC is a what?
Music.
Right. There you go. I actually knew that one, but you cut me off. So yeah, no, Yeah, it's amusing, and this has been previously correlated with survival in adenocarcinoma. Fleck, do you know Fleck? Fleck, any takers? Fleck?
I don't know what it is.
Yes. So again, also, so these have been correlated. And so now we have the opportunity to investigate this a little bit deeper. This was actually, this specific slide was, oh, I have a conference abstract submission deadline tonight. Let me make sure I can go to the conference. Let me throw an abstract together. Oh, let me mine my data for a p-value and then submit it in, and then it got accepted to the conference. And then I was like, oh, conference is like 6 months away. Surely I'll have time to finish the analysis by then. And then the morning of the conference, I'm like on the plane putting my slides together. I was like, oh, I never finished this analysis. So, I think that there's an opportunity here to further investigate what the correlation between these sorts of mutations and the interaction with mast cells is. So we have the opportunity here to continue these investigations in a larger data set, to combine them with methylation. I haven't even touched on methylation yet today, but methylation of circulating DNA has been shown to be a really, really powerful predictor of all sorts of clinical outcomes. Is anyone here doing methylation work? Do you know? Oh.
I used to.
Oh,
but we can still do it if you have the data.
Yeah, well, yeah, we've got the data. Circulating inflammatory biomarkers on these same patients. We have blood, and we're hoping to measure some of them. And then also the interaction with radiomics and what we can learn from a CT scan. So with that, I want to say thank you. This is the picture of us in the mass lab. So again, as I showed you at the beginning, this was Pierre Massillon's lab. When he passed, which was 2 years ago, last month, 2 months ago. Oh, wow, that's insane. We ended up taking what was his lab space and his lab group and reorganizing it into what we call the mass lab. And mass, we preserve some of his name, so it becomes Massillon, but then also it stands for the multidisciplinary approaches for the strategic treatment of lung cancer. Lab, yeah. We never fully got what that was going to mean, but it's close enough. But so anyway, so this is the great thing about this team is that we have multidisciplinary and we have a lot of clinician interaction. Samira Shoje is one of the world's experts in pleural diseases. Eric Rogan, thoracic surgeon. Fabian Maldonado is a interventional pulmonologist. Rafa Pei is a pulmonologist. We have nurses. We have epidemiologists. So the ability to have such a diverse group really allows us the opportunity to take this data that has such a breadth and depth and really ensure that it stays rooted in the clinical context, how we can use that to inform patients. So I did leave a few minutes for questions. So, but also I'm leaving a week for questions because I'll be here until next Wednesday. So if anything I showed you here today, if anything on this buffet was interesting to you, if you would like to, discuss more about what the options for collaboration would be, I'll be here. I sound like a comedian. I'll be here all week. So please reach out. My email and stuff is here. You can also find me on TikTok where I make, the current trend on TikTok is people say DJs will put anything before the drop and they have like a electronic EDM build up and then they say the dumbest thing and then the drop hits. And so I had a couple where I'd have a build up and then announce the title of an abstract from the American Thoracic Society you mentioned and then hit the drop. And that actually got more interaction than any paper I've ever, 30 seconds on TikTok and much more than anything I've spent months or years of my life on. Please connect with me this way or connect with Bear and she can connect us. With that, thank you.
When the vision surrounds